The third and final part of this presentation covers mean comparison tests. The AOV analysis does not provide an answer about which observed differences in treatment means are significant, just whether there is evidence that a significant difference does exist. So the next step of the analysis is to run a mean comparison test. Also known as a mean separation test, we compare treatment means to determine what differences are significant. ARM has a variety of mean comparison tests to choose from, for different situations and research types. The most common ones are LSD, Duncan's new MRT, Student Newman-Keels, and Tukey's HSD. We must also choose a significance level, or alpha, to run these tests. This is similar to the p-value for confidence in AOV results, but sets the threshold for significant differences for the test. Thus an alpha of 0.05 implies that we only accept differences that have a probability of 95% or higher of being true. This selection should be made prior to running any analysis, as the choice should not be biased by desired outcomes. Tests can be classified by how liberal or conservative they are. This means that tests can be more likely to report a false positive, a type 1 error, or a false negative, a type 2 error, respectively. The first four tests listed in ARM are arranged from liberal to conservative. The liberal LSD is the most likely to report differences, but suffers from higher false positive rates. Alternatively, you can be most confident in differences that the conservative Tukey's HSD reports, even though separation is less likely to be found. When deciding which to use, following industry standards or conventions by discipline is a good starting point. Or the study sponsor may request a specific test and alpha level. The purpose of the study should also be considered. When the trial is used for registration, then a conservative test is appropriate. But a screening trial is all about potential, and so a more liberal test may be preferred, because any false positives will be weeded out in future studies. Finally, consider the penalty of failure of the treatments. For example, the failure of a crop protection product will likely have an impact on yield, and so an alpha level of 5% is common. But for a biological product for plant health, the penalty of failure is just the cost of the product and so has a much lower penalty of failure. So a 15 or 20% significance level can be appropriate. Here's an example of how a mean comparison test works. The specifics vary for each test, but the basic concepts are the same. First, a test statistic is calculated to perform the comparison of means. In our example, we will use LSD at 5% significance level, which is 5.49 for this data. Then, compare the largest treatment mean to each of the other means, using the test statistic. If the difference between means is smaller than the LSD value, then they are not considered significantly different. To visualize this process, we can use a number line. The LSD value creates a range around each mean, and anything outside that range is significantly different according to the mean comparison test. Let's start with the largest mean, and extend to the lower bound. All treatments within this range are not significantly different, so we assign the letter A to treatments 5, 1, and 4. Repeat this process for each mean. There are no new means in this next range, so we don't need a letter here. The next range does include treatment 2, and so treatments 4 and 2 are assigned letter B. The next range includes treatments 2 and 3 and is assigned C. There are no more means without a letter, so we are finished. The letters are typically reported to the right of the means. Learn about the reporting tools in ARM that are used to perform and present this analysis in the AOV Report tutorial. In summary, the AOV analysis breaks apart the variance of observations into sources, then calculates a p-value probability. This is the probability that observed differences in treatment means could have arisen by chance. Then we perform a mean comparison test to determine which treatment mean differences are significant. But we must be cautious with this analysis. If the data does not fit the assumptions, we cannot rely on the results. Therefore, steps like data confirmation and data review are important prior to analysis and reporting of results.